Thank you everybody for coming today. Thank you for those who are coming in through Skype, and welcome everybody. It's absolutely fantastic to see you all here, and having received all those data sets in the last few days or few weeks, the response has been tremendous. So it's a really exciting time. Let's go through today's events and a few introductory words about DANIO-CODE, and then I'll pass the word on to other contributors who will discuss the details of the annotation and also give some ideas about planned papers. So the first thing we would like to briefly discuss is communication today. So you already received email invitations and Skype invitations to participate by text, and I hope that you can all see the presentation also by streaming. Now, I believe the streaming will have some delay in it, so your questions and feedback will only be received by Skype text. Please make sure that microphones are off. I believe maybe it's not even possible in this version of Skype to be heard, which is good. But we want to hear from you, so please send your comments and suggestions throughout the presentations, and we'll try to organize question and answer sessions in each slot and answer the questions or provide feedback as we go along. If you have any problems with the streaming, then please contact Bernard Pierce by email, and hopefully it will be sorted out. And the next thing is about the timetable, which is now shown on the slide. And after my brief introduction, Boris will have a few words about exciting news, which we only received yesterday, about funding available for our activities. And then Carsten will introduce the Data Coordination Center, its logic and its structure. And then we will have a 1-hour discussion, which is composed of short presentations towards planning our first paper. And everybody through Skype are invited to send comments and questions and feedback on that as well. After that, we'll have a short lunch, a bit of a break, and rejoin later at 1:15 Central European time, when Matthias will introduce the annotation itself and guide you through in detail how to annotate your data sets. And then onwards from 2 o'clock, we will hopefully get on with the annotations, and if everything goes well, it will be relatively short. Everything is Central European time. So my brief introduction, some of this will not be terribly new, but I think it will be useful to go through it because these presentations will be available on the website and on YouTube, so other people can see it and join us. So therefore, I need to introduce what is DANIO-CODE. And DANIO-CODE has a mission statement, which is on our website, I will show later. And the idea is that we would like to generate a resource which zebrafish users deserve and currently don't have, which is a standardized data coordination center and track hub with all the epigenomic and transcriptomic data sets and variety of next generation sequencing data sets which have been produced so far but are difficult to access. This will improve our visibility as a community, model user community, as well as will be a tremendous resource for genomic scientists in other model systems, and also useful for a variety of other disciplines. Also, we would like to function as a true consortium which shares data ahead of publication, which will be a second phase. And thirdly, we would like to generate data together. So for now, we're focusing on the first, which is the generation of a resource, which will be the track hub and re-annotation, re-analysis of published data sets. Why zebrafish? I don't need to explain that to this crowd, that this is an important vertebrate model. And if you look at these trends of publications, which was produced by Derek Sample, simply looking at PubMed publications, it's pretty clear that among the key animal models, DANIO is the one which has an upward trajectory indicating its increasing importance and popularity. And now maybe 1,000 laboratories in the world are using zebrafish in the lab, and probably a lot more labs using data produced from zebrafish. And it's increasingly used in a variety of disciplines. What is the problem with zebrafish is that, unlike other model organisms, its genome is poorly annotated. And this figure is from one of the reviews, I'm not sure which it is, it might be Dan's actually, is it yours? Which describes a variety of chromatin structures and functions in the genome which have been very well annotated in key model systems such as the human by the ENCODE program, and the mouse with the mouse ENCODE, and in other animal models such as the C. elegans and Drosophila by modern code. But where is zebrafish? And this is why DANIO-CODE has been formed by a group of enthusiastic users to carry out a similar ENCODE style program to annotate our favorite animal model's genome. And our ambitions can be maybe classified as three. As a minimum, we want to have a track hub and generate a platform for information exchange and coordinate published data and make it available to all of us. And where I put yes, that's where the partners have already agreed that we definitely want to do that. And also what I would call the modest ambition is to data share prior to publication has also been agreed, and we will need to generate some sort of an instrument for making it indeed possible to share data prior to publication. And finally, we would like to be ambitious enough to generate a fully blown functional international consortium which will work together in the future to generate data and identify new biologies using zebrafish genomics. Why now? I mean, it's clearly very timely. Now many labs are producing data sets with the most advanced technologies using an animal model, which provides the opportunity to study development in detail. And therefore, it's something which is now a hot topic, and key and major publications have appeared recently in the last couple of years, I say, with zebrafish and providing new insights, genuinely new insights into developmental epigenomics and genomics. We have reached a critical mass. We are a large number of laboratories using the zebrafish, and we need to retain our competitiveness and fundability, for which we need to generate these kind of resources. And we believe there are lots of long-term benefits for generating a zebrafish genome annotation. I listed here a small number of various biological problems where zebrafish has already and will in the future contribute significantly to generate new knowledge. And clearly, one of the most obvious is development, and particularly early development, and the temporal resolution what the zebrafish has provided in identifying the epigenomic, transcriptomic, and other features of early development already, which have important implications, for example, in stem cell biology. As we know, the way how zebrafish starts its genome activation is very similar to the regulatory processes taking place in embryonic stem cells. So it has a broader relevance. Clearly, it has contributed already to epigenetic regulatory mechanisms during development. Zebrafish, as other teleosts, has gone through an extra genome duplication event, which led to some of the genes being present in 2 copies, and that it brings interesting opportunities into studying genome organization and genome evolution, where zebrafish genome annotation will be tremendous value. Genome size is another issue where zebrafish can contribute through comparative genomics and understanding the topologies and the relationship in cis and trans between genomic regions when compared to, for example, human or mouse. Toxicogenomics, epigenetics and environment are key topics today, and that's where zebrafish again can help as a major toxicological model system. The mutations which have been generated by the Sanger and other mutagenesis centers now provide half of the protein coding genes available in mutated form. So this will again be a tremendous resource to combine with next generation sequencing technologies to understand function and functional genomics in general. There's lots of opportunities for brain and studying brain function using zebrafish, where you interface with transgenesis technologies, which can be done in high throughput in zebrafish, and automation technologies, and provide a high throughput complement to genomics data sets. And last, but obviously not least, a variety of disease models zebrafish has provided and will be able to provide in the future. And for this reason, again, its genomics is crucial to develop further. Here, there are some examples of discoveries which have been made using zebrafish and zebrafish genomics. I think I probably won't have time now to go through all this, and really just listed them, and you all know who contributed to these discoveries, and along these lines we would like to continue in the future. When we need to compare how zebrafish genomics looks in comparison to to other model systems such as human or modern code, we really don't know yet how large datasets are. Here are some numbers from previous ENCODE projects. As you can see, there are thousands of datasets which have been used in accumulating the data and then processing and analyzing the data in all of these ENCODE projects. Regarding DANIO-CODE, I'll have a slide in a minute where we see where we stand in terms of published data, and we'll be able to make some comparisons. So how did we start? There was a consortium formation meeting in London, which was supported by a variety of sources, including European Union programs, and the logos are seen on the top, where maybe 25 or 30 laboratories were represented from all around the world, and we discussed how we could start a DANIO-CODE program. And what was discussed is listed here: how we're going to use annotation technologies, what kind of data processing and data sharing instruments we will be able to design and implement, visualization tools, how we're going to network, how we organize the consortium, and who does what, and how we try to publicize our activities. And a lot of this, which was now almost 2 years ago, or a year and a half, I guess, is materialized, and a lot has been done. We did agree on the first level, the phase 1, I call it now, it's level 1 here, which is the generation of a track hub, which is linked to any of the favorite browsers people can use. And this is where we are now, and this is what we're trying to finish off today. And then we will have additional levels with new published data and unpublished data sets. What has been done? A meeting report was written by a group of people, which was published in Zebrafish. The ZFIN established some of the browsers used for genomics, and including Zembu, which is particularly useful for cage data sets, and established a track hub. And we need to thank Anne Eagle and Monty Westerfield for that particularly. In the meantime, a data coordination center was designed by 2 laboratories, Carsten Dow's in Karolinska and Ben Brown's in Berkeley, and we'll come back to that in a minute. We have a website and an email server, and we have a wiki where all the information regarding DCC and any DANIO-CODE related activities are updated regularly. We have recruited people who will work with us on this program, partly funded by the ENCODE IT and International Training Network with lots of PhD students. And just recently we obtained a grant from BBSRC dedicated to DANIO-CODE activities, and it's called DANIO Peaks. And Boris Lenhard will talk about it in a minute. We, of course, need to carry on raising cash for future endeavors, and the opportunities are not a lot, but there are, and I listed a few which I can think of, focusing on the UK, but people should also look at national opportunities for funding in their respective countries, or look at international opportunities and contact us with ideas how we can target these funding sources. As mentioned before, and I guess most of you who are present today, either by Skype or in person, are already familiar with the wiki, which is hosted by Anne Eagle and is regularly updated and contains lots of information about the DCC and the published data set so far. We also have a website, which again, you all have access to, which provides additional information. So these are the data sets which we have received in the last, say, couple of weeks. And it's really fantastic to see the diversity of data types which have been submitted on hard disks or were uploaded. About 2 terabytes of data has been received, representing around 6,000 data files. We're not sure how many experiments that actually means, but we will see that as the annotations go along today, and we will have a better understanding, and this will be updated in the wiki. Again, I'd like to emphasize the fact that this includes a variety of data sets from RNA sequencing, RiboSeq, MNA-seq, 4C, ChIP-seq, variety of types of methylome-seq, CAGE-seq, ATAC-seq, you name it, which is fantastic, and a lot of it is developmental. So we have a in-depth analysis opportunity for vertebrate development using these varieties of data sets. what we would like to do, this is a slightly complicated figure, but it gives you an idea what will happen with these data sets. So this is where we are now. We received all the raw data, the hard disks, at ZFIN, and these data are stored there and will be available on the Trek Hub. Now the next thing to do is, because these data sets were originally in their respective publications annotated on the previous version of the zebrafish genome, it needs to be remapped onto ZV10 and need to be reanalyzed by standard protocols to make them more comparable. And to aid in that, the DCC, the DANIO-CODE Data Coordination Center, has been established by Karsten's and Ben's groups, and Karsten will give us more details about how it looks and how it functions. Today is the Jamboree. Using the arrow here, using the wrong place. When we annotate the data sets, and then once this is done, we can then send this off for processing and remapping to GRCZ10, the latest version of the zebrafish genome. And this reprocessing and remapping will be done by ENCODE pipelines, at least for those data sets for which ENCODE pipelines exist. In case of those data sets which have been produced by technologies which were not dealt with by ENCODE, we will need to agree on the standardized processing, probably using those labs' processing standards, which were the main data producers. Once that has been done, these secondary tracks can be generated and sent back to Oregon for the Trek Hub, and can be visualized by any of the popular genome browsers, by anyone in the scientific community. And that will be the end of phase 1, and this is what we would like to achieve still within this year. So what's missing to achieve all that? Well, we've got the raw data, and today we do the annotations. We are hoping to still receive a few more data sets, for example, small RNA libraries, and a large-scale RNA sequencing project is ongoing right now in Sanger Center. We'll briefly introduce in a minute, by Elizabeth Bush's group. And hopefully there are some others which we may have missed and we need to track and very quickly get onto the ZFIN Trek Hub server, and then we need to introduce some kind of an embargo and stop submissions until the processing has been done and the next phase can be started. So just very briefly, and I hope Elizabeth agrees to this, that I'm showing her slide she sent me a few days ago, which is giving the outline of that rather large RNA sequencing project they are carrying out right now, with 5 replicates for a large number of stages of early development, all the way up to day 5, and with some detail regarding the availability of the data soon. So I'll be hoping that this can be also included in phase 1. And finally, I would like to acknowledge a number of people who have helped to get this far, particularly Monty, who kindly offered ZFIN resources to host the Trek Hub and the data sets for now. Also Karsten, Ben, Marcus, and Matthias for the hard work on the DCC. Shelley for working on the website and registering participants and general communication. Julia, Horsfield, and Matthias for the logo. Fiona for the very tiring coordination and lobbying work she has been doing for years now. And also our funders, ENCODE ITN and BBSRC, particularly Fiona and Boris, who were instrumental to getting these funding. And we would like to also thank Bernard for organizing the Jamboree today. And also would like to thank all the data producers, and I don't have a full list here, who participated through sending the data, which is on the right-hand side of the slide. And thanks in advance to our ZenCode students who worked very hard with phase 1 and phase 2 data. And back again to the timetable. So I pass it on now to Boris, who will say a few words about this exciting new opportunity funded by BBSRC. And this will be the end of the first presentations.